So that actually.
This meeting is being recorded.
Oh, okay. I'm back.
Yeah, you stopping would kicked us all out of it.
Oh, sorry. Oh, yeah. That was
But
what
you sound better now.
Yeah. I think it was my internet that was having problem. I switched to a different router. Yeah, that is this is the same thing I was gonna mention as well. So last time when I wasn't able to make it, I think I was trying to make like other people's hosts, but I can only invite hosts from people from like use it like with like use it email ending. So I can't actually like add Wendy, like you, Wendy, or Matt into, for example, that meeting. So I'm not sure how we're going to solve that. We might need different links or something else, so that, you know, if say if I can't make it, then someone else can record or someone else can use the link or something like that.
Yeah, I don't have a Zoom account in our university. I think Matt has, but that might work. And otherwise, I can make team invites.
Yeah.
But I can only make them, I think, with an. Oh no, I can copy the link, I guess.
So we might need like a backup link or like two backup links, for example, just in case.
Yeah, but the Zoom actually works without you being present, so that worked.
Yeah, but I can't invite anyone to become like co-host or like something like that, or like, so could you actually record it?
No, that's why there wasn't the minutes.
Yeah.
Due to technical difficulties, because we cannot record if we're not the host.
Yeah, so I can't make anyone else's host except for like Kimberly, because she has like a use it ending in her email as well. So that was the problem.
Yeah, and I think the same problem is with Matt probably as well. With his account, he can make Dimitri host, but probably not any of us. And in most of the universities, it's only two persons per union. So that means that will always be the case.
Yeah. But if we could make like one, say, for example, Matt can make a Zoom just like as a backup, so he can run that. Or maybe you can make a team one, just so that we can record it. That's the only thing. Just as a backup. We can still use this as a main one. So I'm gonna still be here for a while, so we can use this for a while.
Okay, then. Well, it's 1:05 here, so I guess everybody who's able to attend is currently in the meeting. Matt already mentioned that he was at a conference and he couldn't make it because he was giving a presentation at the exact same time. I think Dimitri is in that conference as well. I'm not sure, but I don't see him here either. For the rest, well, you are there from University of Sydney. I see that Bernard is there. And from our center, we have me, Ma, and Mimi. If I'm correct, Mimi doesn't have a camera. Mimi, can you unmute yourself?
I'm sorry for Mimi's earphone doesn't work now, so maybe.
Okay. Maybe she can just say hi using your camera.
Hello, hi, I'm Mimi.
So Mimi, can you tell everybody who you are and what you're doing currently?
Yeah, so I've just come to the end of my 4th year of medical school at the University of Exeter, and I'm here on an 8-week internship, and I'm screening some of the new compounds, some of them from London, some of them from various other universities, and some of the sort of more natural perfume compounds we've got.
Okay,
and
thank you.
Mimi will respond for the new compounds recently come. So yeah, I will.
Yes. So yeah, just to sum up, because it was a little bit faint, Mimi is helping us screening all the compounds sent over in the last couple of weeks, months, so that we can screen the compounds in vitro simultaneously with the in vivo work Ma is doing. Because due to the workload of the in vivo model, Ma is not able to do both at the same time. So that might speed things up a little bit. So the agenda, I don't know if you all can see the agenda on GitHub, but I structured it to this, each series. So first, maybe it's good to discuss series 1, because series 1 is of course the series we're the furthest along with. So based on the recordings, well, on the minutes from last meeting, I had 4 action items. And the first action item was for Hang and Dimitri. And it stated that you would both go through the EpiChem library, look up compounds which could fill in the hole of the SAR. And the link of the library was stated in the minutes as well, and I put it in the agenda as well. So Hang, could you give an update on that action item?
Yes, so I've got a link already in the, not the previous one, the one in May. I did make a list already. I actually haven't got a chance to actually send that over to my team. There should be a link in there, and then I will update that in the current meeting. So I think I've got, just give me one moment. I can actually show it here right now. 1 second. Okay, anyway, I can't see it, but I did have a list already. I will update that, and I'll just send that over to my team so that we can get some of the compounds over.
Okay, yeah, I think that the idea discussed last time as well was that Dimitri would go through the EpiChem list as well, so that we could make one order for EpiChem, because otherwise I don't think they would be happy if we ask for one set of compounds and then 1 week later for another set of compounds.
Yeah, so I have that for myself already. So maybe we'll just catch up with, or sorry, follow up with Dimitri and Matt when they have theirs as well.
Okay, yeah, then we keep it as an action item for now so that we can remind Dimitri and Matt.
Yeah.
And you can share your list. Or maybe it's good not to share the list yet with Dimitri so that they can do it independently. Then the next action item for series 1 is that Ma tested and completed some of the fenrimol analogs in vivo. And Ma also updated the spreadsheet with those values. We did that this morning. Although we do have the updates of the centers in the end, maybe it's good now to share your screen, Ma, and present the data from the in vivo screenings, because now we're discussing the fenrimols, and the most data are from the fenrimols.
Okay. Can you see my screen now?
It's coming.
Yeah, okay.
Yes, it is there. Please go ahead. Slide show. So
this is for this month's update. First, we have a small summary from the compounds this year, and the compounds selected from this summer, all of the 15 compounds, and 3 compounds selected from last summer. Yeah, as for these 4 compounds from series 1, they all have done 3 repeats, and I calculated the survival curve. In this figure, we set the ampersand B treatment as the positive control, and the infected control is without any treatment. Then we can see as for these 4 compounds, there are no difference with them. Then I updated the survival in day 10 with log D data, and we found that there are no significance with log D below 2.5 and beyond 2.5. Yeah, and as for the other 2 compounds from series 1, they are the data of molecular weight and log D and rotor bonds and the flexibility and log P should be added. They are still missing. In the other ones, just repeat 1 time. I make the figure 2, and yeah, in the same way, we use the infected control without any treatment, and we use the ampersand B treatment as a positive control because it just repeat 1 time, so there are no significant calculation. As for the series 6, this one is from last summer, and it repeat 3 times. This 5 compounds from series 6 we selected this summer, and just 1 repeat finished. So yeah, it's the same way to test. The infected control is without any treatment. The ampersand B is a positive control. Then we make the figure. As for the nature products, there are no new updates for nature products this month. So this is a small summary. No updates are for nature products this month. And as for the 3 compounds from last summer, 3 replicates were finished, and the survival data has updated in the spreadsheet. As for the 15 compounds from this summer, some data still need to be added, and the therapeutic efficacy tests are still ongoing. As for the new compounds which we received recently, for nature products, we received 35 compounds in liquid, 11 compounds in powder, and 5 compounds from Dimitri, 17 compounds from Andrea, 3 compounds from Kimberly. And the new student, Mimi, will test them soon. Yeah, that's all. I think it is.
Thank you, Ma. Other questions for Ma based on this these data? Ma showed that the log D, I'm just going to see if my computer will open a figure.
Yeah.
The log D, this correlation was no longer valid. We could only add the compounds for which we had a log D, and it appeared that not all log Ds were calculated by Ben, especially not for the new compounds. So I will ask Ben to do that. But I noted that especially the compounds from Dimitri lacked the smileys, and I guess Ben needs the smileys to be able to calculate the log D. So I put it in the agenda, and I will ask Dimitri to upload that data as well. But this is a simple Mann-Whitney test, and I'm not sure if this is the true method to use. But if I share my screen, we have another figure as well. So if you see this figure, the red dots are the azoles and the black dots are all the Fennery moles. And you do see, if you look very carefully, that there's still a correlation like this. So the ones with the smallest log D have the highest percentage survival in general, and with a higher log D have a very low percentage survival. So I will also ask Ben if he could take another look at this data and figure, because he was the one who calculated the significance last time in the first Fennery mole paper, and I think he uses different statistical analysis than we did. Because if you look at this, I would say that it looks like there is still a correlation there. But yeah, I don't know what the chemist opinion is of this, because yeah, for me, it's just looking at the graph, what I would do if it was a biological graph. But yeah, so are there any thoughts on this one?
Yeah, I think so it could also be as well that log D is just one of the parameters that will determine that, but I would say yes, that I can see the trend, but I'm also not sure like how like if there's a more statistical way that we can actually determine, like the other one that Ma showed before as well. I'm actually not sure which one will be the best way to actually say definitely or not that there's a correlation, but yeah, on this particular graph you can actually kind of see that as well. Like I do agree with you.
Bernard, you also unmuted yourself. Go ahead.
I have a different question. What have you used as a positive control in your in vivo tests? Was it amphotericin or another azole like?
No, we used amphotericin B because it always. Oh, Mark would like to answer. So Mark, go ahead.
Yeah, we use the amphotericin B as a positive control. Yeah, we test many compounds inhibit the fungi growth in vitro, like intraconazole, terbinafine, and amphotericin B. But we found the amphotericin B works in larvae very well. So we selected the amphotericin B as a positive control treatment.
But because the most test compounds are azoles, it might be interesting to see if there are differences or improvements in the existing compounds compared with itraconazole, for example.
Yes, so in this graph you see some red dots. That means that all these red dots are different azoles. Mm-hmm. So we actually did the same trick with the azoles. But we didn't find that high survival with most of the azoles. This one is ravuconazole that works the best. Mm-hmm. Posaconazole, itraconazole. Well, itraconazole doesn't work. Posaconazole is dose dependent. And for this comparison, we could only use those azoles at the same molar, because otherwise it wasn't a fair comparison. Mm-hmm.
Maybe I'm wrong, but can it be that some of these azoles were quite close to the graphs of the amphotericin sample in your graphs you showed?
Those were the Fennery moles, but since Mark only repeated the compounds which did seem to have some activity, she only repeated once. So usually we only do the statistical analysis if she did repeat it at least twice, 3 times.
And so activity means then it must be better than amphotericin B, or is there a different criterion?
Ma,
it has
could you answer this
significant,
one?
yeah, it has significant with the infected control. Yeah, if the compound has a significance with the infected control, then we set it as the positive, the promising one to inhibit the fungal growth.
Okay.
Yes, so we compare it with the infected control. We always include amphotericin B just to make sure that there's nothing wrong with the experiment. So the way we read the experiments, because sometimes it's tricky to work with the larvae because they are also influenced by the weather. So an experiment is only valid when the infected control dies within a certain amount of time, so within the 10 days. The non-infected controls don't show any deaths or only 1 or 2, so they stay on top. And amphotericin B prolongs larval survival compared to the infected control, because then we know that all the conditions were as they are supposed to be. If there are no other questions on this part, then as mentioned before, that's the third action item of series 1. We do miss some log-D data and some smileys. So please, Hang and Dimitri, check the spreadsheet to see if the smileys are present. I will approach Ben to see if he can calculate log-D. I will do that via GitHub, but also via email, because usually he responds quicker via email. And of course, we're still drafting, that's the fourth action item, the manuscript. Are there any updates on the manuscript, Hang?
Yes, so the I put all of my data, chemical data characterization onto the draft now, as well as the essay, the most updated version of the essay. So that part from me is done as well. And I saw that Dimitri also put his data on already. So I think we are done with the like the updating the data, chemical data as well.
Okay. Yeah, Wilson made a version for his thesis, and that one is submitted for the plagiarism committee. That's one of the requirements for a Dutch thesis. I will go through the manuscript one more time after my holiday to check the introduction, discussion, etc. if those two versions are almost simultaneously. Because I know there have been some changes here and there in that manuscript. And of course, we are still waiting for the last results from Ma to be able to enter into the manuscript.
I think Kimberly back then, she actually made a separate, smaller document that contains only in vivo compounds that was supposed to be just updated for specifically for Wilson to use. Does he still need that one, or is the manuscript okay? Because I put my data onto both of those.
I think that's okay as well, because he made a draft version into his thesis, so he doesn't need to be on the GitHub. For now, when it goes to the reading committee, it stays as it is in his Word document. And he can update it later after it has been through the reading committee, if he needs to.
Yep.
Then we have series 2.
Sorry, before we move on to that, what is the next sort of like things that we need to do after? Like we've already got all of the stuff onto the draft. What do we need to do afterwards?
So now it's just, I think for Matt and me at least, to go through the manuscript to see what is still lacking. And we have to wait because there's no use in submitting this manuscript if there are a few more veterinary mole analogs to be analyzed this year. So last time we said we make it as far as we could go now to complete everything with the chemistry, etc., the in vivo, in vitro. And the last in vivo data, which will be derived from this season, will be added before we submit. So it might be that, like for instance, if the analysis with the log-D or any other variant, like rotation bonds or molecular weight, those things need to be updated. And maybe Ben has some other software where he could look for the for if the association still stands. Because with Man Whitney it doesn't. But maybe if you look at with a different statistical analysis, it still does. And of course with the SAR, so that's why we would look into the SAR to see if there were some obvious missing links which would already be in the EpiChem library, and those we would test as well. And those were the last action items for the manuscript as far as I knew.
Yes. One other thing is what that I can do is I can actually ask my supervisor, so ask Peter Rutledge and also Alice Motion as well to double check, because I think they would like to just help out as well and be one of the authors on the paper. So I can ask them to just also go through as well, like while you Wendy and Matt also having a look at it. So we have multiple people just looking through it, just make sure everything's okay.
Yes, that would be great. Still other things on the manuscript?
Okay. No, that's all from me. But also before we move on, I did send on behalf of Kimberly. I sent 3 compounds. So I saw my updated that. I did not include any chemical data like the smiles and the names and stuff, because it's actually Kimberly's compounds. I'm gonna go and ask Kimberly to actually send that over to you guys. It will be faster for her to search through her own work to just send the data over. That's why I only sent the compounds and the names. So I think it was 38, 39, and 40. That was from Kimberly. But I'll ask her to update the data with you.
Kimberly has updated the data for
Okay.
3 compounds.
Okay.
Yeah.
So you've got like all of the molecular weight and all things like that. Is that right?
Yeah.
Okay, that's good. Yeah, she's no longer like working as postdoc for UCET anymore. So like right now it's just back to just me again. So yeah.
Yeah, we contacted the Kimberly at that day. We received the compounds.
Okay.
So she updated the data for us.
Okay, that's good.
Thank you.
Yeah,
Okay.
that's all from me. Oh, sorry. One more thing.
Yes,
I have a lot of things today. I also went to the RACI 2022 Congress. This was like the Royal Chemical Society of Australia. Basically, we're just a lot of chemists coming together to like just showing our research and stuff. So I actually had a chance to present some of our work at RACI as well, which was really a lovely experience as well. It was my first in-person conference. So being able to just share, I think it was, I shared the Phenaromals, so the series 1 work at RACI, and Kimberly actually shared her work with high schools and undergrads, so for series 2. So we actually have some representation at RACI as well. I think Matt is not here today actually, but I think we've met one of Matt's collaborators on open source malaria. He was actually interested in sending some compounds over. So I'll actually see if I can reach out to that person and see if he can send some compounds over as well, if he's still interested. But yeah, we shared some of our work. We shared our GitHub. Hopefully, I don't know if anyone was interested enough to like join us in the future or something like that. It would be great. But yeah, overall it was really a really nice experience. So I just want to update everyone on that.
Yeah, that's great. And I saw that there's still many people more joining in GitHub. It goes slowly, but it does. We do see more people coming in. So I think advocating for MycetOS on your end, on my end, on Matt's end, and also Bernard reached out to several people. It does help.
Yeah. So yeah, hopefully we'll get more, more result, and we can also do things faster as well. But yeah.
Yeah. Yep. So then, since you already mentioned, yeah, since you already mentioned that Kimberly spoke on the meeting as well, that's a nice bridge to Series 2. So Kimberly sent some compounds to us, and the action item from last meeting still was to update on Series 2 work with the high school students and check if there's no doubling of compound synthesis. So maybe she can check that with Matt later on. Then there's no updates for Series 3 and 4. I think they are currently on hold.
Yeah, concerning Series 3, the phenothiazine, I think Anthony asked me for chlorpromazine hydrochloride. And I have that in stock because we used it as an endocytosis inhibitor for drug uptake and so on, drug uptake studies. And of course, I can send you this compound if there's still interest. It's a pure compound, yeah, chlorpromazine.
Yeah, we can take that along, especially now Mimi is screening a lot of compounds. I don't think one more or would hurt much. So yes, please, if okay, you can send it over, or if when a next shipment from your side comes in, just
I think I think I read that you already tested methylene blue because it's also quite similar compound, and maybe there's some similarities or differences, don't know, in the activity.
Yes, we did test that in the past, indeed.
Mm-hmm.
So with respects to Series 5, you sent an email to Ben at DNDI. And based on that email, we received some 17 compounds from Andrea Otego. At least that's the name written down in her GitHub stream. And we were also approached by somebody else who had other compounds with the same chemical backbone. I'm not sure how many compounds there are from that person, but of course we can take them along as well. So I think that compound series is growing as well, so that's great. Any updates on that? Do you would like to add something, Bernard, on that?
No, that's great. That's perfect. I think we can see then if there are more active compounds, different structures with activity and so on. It's perfect.
Okay. And for Series 6, Ma already updated that she screened some of them in the larvae and that the final screening still need to be performed. And for Series 7, Ma and Mimi together made a library. So all the compounds which did came in were put on 96 well plates. And well, Mimi didn't have a microphone, so maybe Ma, you could update on that?
We made a list for the the series 7 compounds, but the final version is in Mimi's side. Maybe we can share it after the meeting or put it on the GitHub website.
Okay, that's great. So all the compounds we recently received, they were made in one big 96 well plate, well, 2 big plates, yeah. And they will be screened in the upcoming months. And Mimi is here for 8 weeks, so hopefully by the end of the 8 weeks we have screened everything, not only at high-low concentrations, but the most promising also for IC50 and maybe even MIC50. But if we go all the way to, if we finish IC50, that would already be great. And then by the last part of the meeting, we have updates from each lab. So Hang, do you have any other updates you would like to share, which we didn't touch upon yet?
Yeah, that's all from me. I did have to pause Synthesis in Series 5 because I was just having some issue with Synthesis on that one, and I was gonna come back to Series 1 because there was a few sort of like compounds of interest that Kimberly suggested. So I'm gonna just finish off some of that, and I'll come back to Series 5 just to troubleshoot my Synthesis route. So that's what I'm up to, apart from just doing Raki and doing draft. But yeah, that's it's just a brief update from me this time.
Okay, well, there's nobody from London, so we can't have an update on that. Ma, I think we already gave all the updates we have, don't you?
Yeah.
Okay. Bernard, do you have any updates you would like to share?
Okay. Well, I think that I want to write next month a manuscript about the naphthoquinone compounds. I think that's almost finished with the chuclone and with the naphthoserine and so on. This compound series, I think that might become a nice small manuscript or letter, something like that. That's what I want to do. In terms of compounds, well, I'm waiting for 3-4 perfumes from IFF. Still haven't received them. I think there's a problem with one of the compounds. I told them last week that if it's not available, they should send me the other 3 compounds. But since then, I haven't received an answer from them. But I hope I receive the compounds then in August or September latest.
Okay. Then the last thing was there was this UNESCO call. Matt already submitted it. It was a little bit of a different call than I'm used to. But well, one of the things Matt also mentioned is that MycetOS would run smoother if there was some core funding from organizations like UNESCO. So hopefully that will help. And yeah, with respect to funding, I did do a small grant application here internally at the Erasmus University. We do have a small fund for some seed money. I hope to be able to get some money to continue the screening, because that would of course be very helpful in this project, because our core funding runs out next year. So I hope that by securing a small grant, we could continue with the screening cascade. That would also mean that I'm going to search for other opportunities to apply some funding for this project. So if anyone sees something which we could all apply to or jointly apply to, please let me know. then we could try to assure some more funding to continue the screening. From my side, there are no any other business, but are there any other business from Bernard? Because you're still on,
Yes, Dr. Nwisi from Nigeria contacted me about the shipping process and so on, and we discussed that, and it was also about covering the shipping costs, for example. And I think DNDi has a mechanism or a program that can cover shipping costs from Africa or Asia to your lab to Rotterdam. So maybe that might be something we should keep in mind for the next autumn or winter when they want to send their extracts, plant extracts to your lab, that they can ship it for free, or that the costs are covered by DNDi, for example.
yes, that would also be great, because for them shipping cost is of course tremendous.
Okay, yeah, that's all from my side. Thanks.
Okay, Hang, do you have any other things you would like to discuss?
That's all from me as well. I will also double check if there is any funding opportunities from UCIT as well, but we'll see. I'll get back to you on that.
Okay. Ma, any updates still from your side?
Yeah, I think I show all updates from my side.
Mimi, do you would like to add something? No? Okay, then it was a quick meeting. We finished in 45 minutes actually. Yeah, the next meeting I think is at the end of August. So then I will all see you next time. I think then it's the most convenient time for Hang again.
Sorry?
I think it's the alternating time then again. So now it's here in the afternoon. So then it's for us in the evening. So it's for you the more convenient time probably.
Yeah. I think it's probably like 7 in the morning for us. Yeah, all good.
Okay. Okay, then have a nice summer.
All right, thank you. Have a great week, everyone. Hope to see you soon.
Yeah.
Okay.
Have a nice
Bye.
summer.
Bye.
Bye.